Greetings from Chernivtsi, from Ukraine. We continue a series of lectures for entrance. Let me remind you about myself. My name is Mr. Vladislav Yusupenko. I am the assistant of the Department of Medical Biology and Genetics. And today I will tell you about the life cycle of the cell and cell division. So if we're speaking about the life cycle, first of all, you should know that cell cycle is a whole period of life of our cell, which include a cell division. So cell cycle contains two main phases. So it will be interphase, which occurs in the 90% of the cell cycle. And also we have the M phase, which takes place only in the 10% of the cell cycle. So let's start first about the interphase. Interphase is a period between two cell divisions, and our interphase contains three main subphases. So first is a G1 phase or pre-synthetic phase. So here our cell will just formed by cell division, and they grow and do their normal function and metabolic activity. So, and set of hereditary information in this case will be 2N for C. So let me remind you that N is a number of chromosomes, whereas C is a number of DNA molecules. So I pay your attention that each chromosome have just a single chromatid, so one DNA molecule. So at G1 phase, human somatic cell will contain 46 chromosomes and 46 molecules of DNA. Important is the next stage, S phase or synthetic phase, because at this stage, replication or synthesis of DNA molecule take place. So from this moment, the set of hereditary information in the cell will be 2N for C. So somatic human cells will have 46 chromosomes and 92 molecules of DNA. And each chromosome will have two chromatids. That's why it will be called bichromatid. And the last stage of interphase is a G2 phase or post-synthetic phase. So in this case, our cell prepare for the division. So the accumulation of energy take place, synthesis of specific protein, tubulin protein take place, which are necessary for the cell division. And the set of hereditary information also will be 2N for C, and each chromosome has two chromatids. So after the G2 phase, mitosis will start. So be attentive. Mitosis, it consists of two divisions. So first is a division of a nucleus, which called karyokinesis, which have four main steps. It will be prophase, metaphase, anaphase, and telophase. After the karyokinesis, division of cytoplasm take place, which called cytokinesis. So let's start first with karyokinesis. The first step is prophase. So what will happen at this phase? First of all, spiralization or condensation of chromatin take place and formation of chromosomes. Our centrioles move to the opposite poles of the cell, and our spindle fibers start to appear. So the nuclear membrane is destroyed, the nucleolus disappear, and the chromosomes are freely placed in the cytoplasm. The set of hereditary information here will be 2N for C. It will be 46 chromosomes, 92 molecules of DNA. Next phase is a metaphase. Here, our two chromatids chromosome are located at the equator in a row, forming a metaphase or equatorial plate, making the star. So here, our spindle fibers are attached to the centrosome of chromosome on both sides. The set of hereditary information will be unchanged. It will be 2N for C. So it contains 46 chromosomes, 92 DNA molecules. So I pay your attention that at metaphase, our chromosomes are maximally condensed. So they are the largest in this case. And usually they located in one line. That's why it's very easy for us analyze our karyotype at this stage. I remind you that karyotype is a set of chromosomes in the cell. So most of chromosomal disorder, like a Down syndrome, like a Patau syndrome, Klinefelter syndrome, Turner syndrome, we can diagnose this by analyzing the karyotype during the metaphase. After that will be anaphase. So during the anaphase, our spindle fibers are cut and break the chromosomes into separate chromatids, which become daughter chromosomes. And these chromatids move to the opposite poles of the cell. And be attentive, each daughter chromatid become a separate chromosomes. So at the short period of the time, because anaphase is the shorter phase, so for the short period of the time, the set of heredity information in the cell will be 4N for C. We have 46 chromosomes, which contain 2 chromatids. So they are split. And that's why 46 we multiply in 2, it will be 92 chromosomes and 92 DNA molecules. And the last phase of mitosis is a telophase. So here our chromosome decondense and 2 nuclei begin to form around them. The nuclear envelope around the chromosomes is formed, nucleolus reappears, microtubules of spindle disappears. So telophase is an opposite phase to prophase. And here we have a cell with 2 nuclei, and each nucleus have a set of heredity information 2N to C. So each nucleus will have 46 chromosomes, 46 DNA molecules. After the karyokinesis, cytokinesis take place. Cytokinesis is a division of cytoplasm, but here will be a small difference between plant and animal cells. So in animal cells, they involve formation of contractive rings, whereas in a plant cell, a new cell wall between 2 daughter cells is formed. So let's make a short conclusion. So before the mitosis, we have one maternal cell with a 2N for C. So after all 4 stages of mitosis, after the cytokinesis, we get the 2 genetically identical daughter cells. So we get the 2 cells with 2N to C. So they are identical, genetically identical to the maternal cells. And let me remind you that I have prepared interactive pictures for you to make your biology studying more interesting. You can find these pictures by simply scanning this QR code with your phones. So what about significance of mitosis? First of all, it causes increase the number of the cell. Also, is a basis for the growth and regeneration of any multicellular organism. It saves a set of chromosomes in all somatic cells. It's a mechanism of asexual reproduction in which offspring are formed that are genetically identical to the parental individuals. And of course, it gives us an opportunity to study the karyotype of an organism during the metaphase. So while studying at our university, students have the opportunity to do a lot of interesting practical work to acquire skills that will be useful for the doctors. As Cesar taught, as rectum omnius magister usus, what means experience is the best teacher. Here you can see a temporary microslide made by my student, which showed a different stages of mitosis in onion root cells. For example, this one, it's a metaphase, this it's a anaphase, here it will be the telophase. After that, let's start our discussion about the meiosis. So first of all, meiosis occurs in two divisions. So the first division, it's a reduction division, and the second division is a equation divisions. So I pay your attention that before meiosis 1, before the first meiotic division, it will be interfaced with replication of DNA. So that's why before the first meiotic division, the set of hereditary information will be 2N for C. But before the second meiotic division, our interface will be very short and without replication, or our interface will be absent. That's why meiosis lead to the reduction of the number of chromosomes, which help to maintain a constant number of the chromosome in the species from generation to generation. For example, look at this picture. We have two diploid organisms. They are formed haploid cell by meiosis. So by meiosis, it will be a production of a sperm cell in a man and egg in a woman. So after the fertilization, the zygote receive again diploid set of chromosomes. So 23 chromosomes from egg, 23 chromosomes from egg, they give us 46 chromosomes in zygote. So that's why our zygote will have diploid number of chromosomes. So first we'll start with first meiotic division, which called meiosis 1. The largest phase of this division, it will be prophase, which called prophase 1. So why is the largest phase? Because here the prophase 1 are divided in 5 subphases. So we have leptotene, zygotene, pachytene, diplotin, and diakinesis. So I will pay your attention to the zygotene and pachytene. So during the zygotene, our bivalent are formed. It's a pair of chromosome. And during pachytene, crossing over take place. What means crossing over? Crossing over is exchange of chromatin material, exchange of genes between the two non-sister chromatids of homologous chromosomes, and it leads to the recombination of a gene. After that will take place metaphase 1. So here our bivalents or tetrads are at the equator of the cell, and they form an equatorial plate. Microtubules in this case, be attentive, they are attached the centromeres of the homologous chromosome. So let's compare mitosis prophase and meiotic first metaphase. So here, for example, we will have a mitosis metaphase, and here we have a meiotic first metaphase. So at the mitosis, our chromosome lie at the equator forming a metaphase place, but our chromosome located at one line. So each chromosome located separately one from another one. But in the case of metaphase 1, our chromosomes still will be in the bivalence. So they are stay in the pairs. And one more difference, it will be about the attaching of the spindle fibers. So in the mitosis, our spindle fibers attach to the centromeres of chromatids, whereas at the metaphase 1, our spindle fibers attach the centromeres of separate chromosomes. Let's continue. So the first, after that will be an anaphase 1 phase. So at this case, our chromosome will split and move to the opposite poles. So again, let's compare the anaphase in mitosis and anaphase in meiotic first division. So during the mitosis, our chromosomes split into two daughter chromatids. So our daughter chromatids move to the opposite poles. But in the case of anaphase 1, our bivalent splits and chromosomes which have two sister chromatids move to the opposite poles. So it's a main important differences in these cases. After anaphase 1 will be telophase 1. So telophase 1 are similar to the telophase in meiosis. So in this case, the nuclear envelope around the chromosome reforms and the nucleolus reappears. The despiralization of the chromosomes take place and formation of chromatin, and cytokinesis take place and two haploid cells are formed. So in meiosis 1, one maternal cell with 2N4C produce a two haploid cells which will have set of heredity information 1N to C. So meiosis 1 proceeds directly to meiosis 2 without the replication and without the interface. And the second meiotic division are most the same like mitosis division. And I pay your attention, the main differences will be that the cell have a haploid number of chromosomes, and each chromosome contains of two chromatids, whereas the first meiotic division, it will be a cell with diploid number of chromosomes. So, and after this, after the meiosis 2, so we will get two cells with a set of heredity information, 1N, 1C, and the cell have different heredity information due to the crossing over. So, for example, look at this picture, please. So here we have the maternal cell with 2N4C. So after the meiosis 1, we will have two daughter cells, and the set of heredity information in that cells will be a 1N. So the number of our chromosomes will be 23 chromosomes, but the number of DNA molecule will be 2C. But after the second meiotic division, we get a 4 daughter cells which have the set of heredity information 1N, 1C, which means 43 chromosomes and 23 chromosomes and 23 DNA molecules. So what about significance of mitosis? So of course they maintain a definitive and constant number of chromosome from generation to generation in sexual reproduction. And also they provide a genetic variation by recombination of heredity material. So it's the main reason why all of us are different. So we are not identically like our parents. So we are similar to our parents, but we are different. So the main reason for this, it will be the crossing over in the prophase 1, and also it's a random segregation of chromosomes during the anaphase 1. And in the end, let's compare mitosis and meiosis. So first of all, mitosis occurs in the somatic cells, whereas the meiosis occurs in the gametes. So mitosis underlines asexual reproduction, whereas meiosis underlines sexual reproduction. Mitosis is always one division, and replication will be before each division, mitotic division. Whereas if we're speaking about the meiosis, it contains 2 divisions, but replication takes place only before the first division. So there are no interface before the second meiosis. So also mitosis has no crossing over, whereas meiosis has crossing over. And after the mitosis, we get 2 diploid cells, and after meiosis, we get 4 haploid cells. So this interactive picture, which you may find by simply scanning QR code, will help you in studying the differences between these 2 processes. So thank you for your attention. Dhanyavad. Join to us, become a member of the huge BSMU student family. So take care. See you soon. Fir milende. Namaste, dear future students.